Hello, today we are going to discuss about allosteric regulation. Now, this is a particular type of regulation which is very, very important and very, very happening in our living systems. Okay, so a regulation of enzymes is very, very important, out of which allosteric regulation is a type of regulation. We have different types of regulation of enzyme, and we all know that enzyme regulation plays a very important role because it is a part of every process which takes place in our living system. Now, out of the different types of regulation of enzymes, covalent modification is one type, and allosteric regulation is another type. And of course, we have yet another other types, for example, zymogen, proteolytic cleavage of enzyme. So these are all different other types of regulation of enzyme. But today we are going to discuss about what is called as allosteric regulation. Now, allosteric, the word itself tells that the regulation is happening at a site other than the active site. Okay, so usually your substrate, the substrate and the enzyme, usually binds at the active site. Now, this allosteric regulator's role is not by competing with the active site by coming and binding to the active site, but it goes and binds to a site other than the active site, and that is how it is regulating the enzyme. So that is how it has got this term allosteric regulation, means binding at a site other than the active site. And how does it regulate the enzyme? Now, I'll start sharing the screen. Okay, so today's topic is allosteric regulation. Now, allosteric regulation of enzyme is crucial for the control of cellular metabolism. So it occurs, when does it occur? When an activator or inhibitor molecule. So these allosteric modulators, so they are generally called as allosteric modulators. It can be a positive modulator or it can be a negative modulator. If it is a positive modulator, you call it an activator. And if it is a negative modulator, you call it an inhibitor. Okay, so what does it do? It binds at a specific regulatory site. So as we know, enzyme has got two sites. One is the catalytic site and one is the regulatory site. Catalytic site is a site at which the enzyme and substrate binds together, and regulatory site is a site away from the active site where the binding of the modulators takes place, and thus the regulation of enzymes takes place. So it binds at a specific regulatory site on the enzyme and induces. What does it do? It induces a conformational change. So how does this regulation happen? Okay, so what is meant by conformation? Conformation is nothing but the arrangement of atoms in space, and we all know enzyme is a protein, and protein is having its quaternary structure where the atoms are arranged in a specific manner so that you have an active site and you have a regulatory site. So when this arrangement of atoms moves, the actual function of the enzyme itself can change. Okay, so what happens when this particular modulator comes and binds to the allosteric site or the regulatory site? It actually changes the conformation. Conformation means it changes the way in which the atoms are arranged in that particular regulatory site. So that, what does it do? It can either enhance or reduce enzyme activity. So the binding of this particular modulator, whether it be an activator or an inhibitor, can actually affect the enzyme activity in a positive way or a negative way. Not all enzymes possess sites for allosteric binding. Those that do are called allosteric enzymes. So you cannot say that all enzymes are allosteric enzymes. Only few are allosteric enzymes. So those which possess the sites for allosteric binding are called as allosteric enzymes. And the ligands that bind to the allosteric enzymes and affect binding at different site on the enzyme are known as effectors. Okay, so these effectors can be positive modulators or negative modulators. As the word tells, they are causing an effect, okay, whether it be a positive effect or a negative effect. Now, if you look into this, you can see the structure. Here you can see that this is the active site and this is the allosteric site. And the substrate comes and binds to the allosteric site, not at the active site. So what happens once it comes and binds to the allosteric site? What happens? No, it becomes inactive. So this enzyme has become inactive now. This was active, Once the substrate has come and bound to the, substrate usually comes and binds to the active site. This is the regulatory site. The allosteric inhibitor comes and binds to the regulatory site. So what is happening? The conformation changes, and then the substrate can no longer come and bind to the catalytic site. Okay, so allosteric enzymes have an additional site other than the active site or substrate binding site. The substrate binding site is known as the catalytic unit subunit, which is called as C. And the effector binding site, effector is what is called as your allosteric effector. That site is called as the R unit or regulatory subunit. And always the effector comes and binds to the regulatory subunit, while the substrate comes and binds to the catalytic subunit. Now, allosteric sites are binding sites on the enzyme. They are different from the active site and the substrate binding site. That everybody should be clear. Okay, so the allosteric sites are different from the active site. The molecule that binds to the allosteric site is called as an effector. It can also be called as a modulator, and it regulates the activity of the enzyme it binds to. Now, the activity of the enzyme is increased. When is it increased? When a positive allosteric effector binds to the allosteric site. And this means that the activity of the enzyme is decreased when a negative allosteric effector binds to the allosteric site. That is what is called as an inhibition of the enzyme. So allosteric enzymes are larger and more complex than non-allosteric enzymes and often have many subunits. So that is something different. When you compare with the ordinary enzymes with that of the allosteric enzyme, they are found to be more larger and more complex and has got an extra subunit which is called as a regulatory subunit. Now, enzymes with more than one effector have different and specific binding sites for each one. For example, this effector can be more than one, which is controlling the enzyme's action. Okay, so if that is the case, each effector will have a different regulatory subunit. In most allosteric enzymes, the substrate binding site and the effector binding sites are on different subunits. So, you know the protein structure, you have different subunits. Enzyme is a protein, and you can see that the substrate binding site is in a different subunit, and the regulatory or the modulator or the effector binding site is at a different place. So the substrate binding site is on the catalytic subunit, often referred to as a C subunit, and the effector binding site is on the regulatory subunit, often referred to as the R subunit. And when an effector molecule at one binding site causes a conformational change in that subunit, a conformational change is then caused on the other subunit of the protein. So suppose this is the protein. If the effector is coming and binding here, and if this is the cleft here, this binding causes a conformational change that even the active site conformation changes. Okay, so the binding at one site causes a change in the active site also. This means that a huge portion of the binding energy of the effector is used to change the conformation of the whole protein complex. So what does it do? As it binds and the binding energy of the effector, what does it do? It actually converts the conformation, or it changes the conformation of the whole protein complex, thereby rendering it inactive. Now here in this structure, you can see that suppose this is the enzyme, this is the allosteric site, and this is the active site. Usually the yellow one, which is substrate, comes and binds to the active site. Now what happens when an allosteric inhibitor, though it is binding at a site away from the active site, it switches off the enzyme. Okay, so once this comes and binds, the conformation changes in the active site also. So this cannot come and fit here. So once this binds, it is switching off this particular active site also. So activator increases the activity of an enzyme, whereas inhibitor decreases the activity after binding. So the velocity versus substrate concentration graph of allosteric enzyme is an S curve as compared to the usual hyperbolic curve. This is something which you should remember. Usually enzymes show a hyperbolic curve, while an allosteric enzyme shows a shape of an S curve. So you can understand that the binding of something which is a modulator or an effector to a site other than the active site causes a conformational change in the active site, thereby rendering the enzyme inactive, or the substrate cannot bind to the active site, or the rate of the reaction is affected. Now, under these allosteric regulations, we have homotropic regulation as well as heterotropic regulation. A homotropic allosteric effector is a substrate for the enzyme as well as a regulatory molecule. And as the term tells you, the prefix homo refers to being the same. Okay, so they are usually activators of the enzyme. And homotropic is a good example of allosteric effector is oxygen. For example, when one oxygen comes and binds, it is regulating the next oxygen also to bind. That means the binding of a particular substrate itself is acting as an effector. Okay, so a good example of a homotropic allosteric effector is oxygen. It acts as an effector of hemoglobin in the human body. Now, heterotropic allosteric effector is a regulatory molecule, which is not also the substrate for the enzyme. It is a different molecule. It can either activate or inhibit the enzyme it binds to. So this image shows a heterotropic allosteric effector. Here you can see they both are different. It is not homo. This is a square and this is a sphere, which denotes that they both are not homologous molecule or same type of a molecule. A good example of a heterotropic allosteric effector is carbon dioxide. It is also acts as an effector of hemoglobin, but is not the enzyme's substrate. Now the sigmoid curve, you can see the difference between both. The normal curve is what is called as the a hyperbola, while for allosteric enzyme you have an S-shaped curve, which is also called as a sigmoid curve. So the sigmoid curve is given by the homotropic enzymes in which the substrate also serves as a positive modulator. And for curve for the non-regulatory enzymes, which is not regulated, is hyperbolic, as also predicted by the Michaelis-Menten equation. Whereas allosteric enzymes do not show Michaelis-Menten relationship because their kinetic behavior is greatly altered by variation in the concentration of the modulator. So this does not follow the Michaelis-Menten equation, and hence it shows a sigmoidal curve. When an effector molecule at one binding site causes a conformational change in that subunit, a conformational change is then caused in the other subunits in the protein. This means that a huge portion of the binding energy of the effector is used to change the conformation of the whole protein complex. Now coming to allosteric activation and inhibition. Now this is how the enzyme structure is. Okay, so you have two different sides in the enzyme. One is for allosteric activation and one is for allosteric inhibition. Okay, so allosteric inhibition, when an inhibitor binds to the enzyme, all the active sites of the protein complex of the enzyme undergo conformational change so that the activity of the enzyme decreases. While if you take up the case of allosteric activation, when an activator binds, it increases the function of the active sites and results in increased binding of the substrate molecule. So you have two completely opposite action which is happening for allosteric activation and allosteric inhibition. So in the enzyme itself, we have different binding sites. One is for positive allosteric site and to which the positive allosteric effector can bind. The other one is a negative allosteric site where the negative allosteric effector can bind. And this is another area where you have the cofactor which can come and bind. And this is your catalytic area where the substrate comes and bind. Okay, so the whole enzyme molecule itself, you have different areas for the binding of different molecules. Now, these are different examples of allosteric enzyme. The classic example is aspartate transcarbamylase, which is involved in the biosynthesis of pyrimidine, and CTP is the end product and also inhibits the reaction, and it is also known as feedback regulation. ATP, a purine nucleotide, activates the process. High concentration of ATP can overcome inhibition by CTP. And this ensures the synthesis of pyrimidine nucleotide when a high concentration of purine nucleotide is present. And other examples of allosteric regulation are glucokinase, acetyl-CoA carboxylase. Okay. Similarly, regulation of glycolysis is done by the allosteric regulation of phosphofructokinase. And you can see that here. This is the three major forms of allosteric regulation of phosphofructokinase 1. You can see that AMP and ADP is a positive modulator while ATP is a negative modulator. So fructose 6-phosphate is converted to fructose 1,6-bisphosphate. So as per that, The glycolysis proceeds fast and you have ATP being synthesized. And suppose the glycolysis is taking place in a fast way, and you have a lot of ATP molecules being synthesized. Your body does not require any more of the ATP. So what happens? ATP will come and stop the reaction. So ATP is acting as a negative modulator. While if you don't have enough ATP, you will be having AMP and ADP. So AMP and ADP will promote the formation of ATP. So that can act as a positive modulator for this phosphofructokinase. So this is one of the rate limiting steps in glycolysis, where the different modulators can actually modulate, acting as a positive effector or a negative effector depending upon the need of the cell. Okay, so if the cell does not have enough ATP, AMP and ADP will come and act as a positive modulator promoting glycolysis. If there is a lot of ATP and there is a lot of energy, though, it has to stop. Glycolysis has to stop. So what happens? ATP itself will come and act as a negative modulator. So this is yet another type of a regulation which is called as feedback inhibition, where the product is formed in bulk. So the products itself will come and tell, they give the message, okay, that is enough, you can stop now. Okay, so that is what is called as feedback inhibition. So the final end product inhibits the enzyme by feedback regulation when excess of the products form. So sometimes in your body there are multiple metabolic reactions happening. It can be fatty acid metabolism, carbohydrate metabolism, nucleic acid metabolism. So everything is going on in a very regulatory manner. So the cell itself takes care, and sends signals to each other as to regulate these metabolic pathways. So these are all done by different methods like your allosteric regulation, feedback inhibition, and things like that. So feedback inhibition is where when you have a lot of product form, the product itself serves as an inhibitor. It goes and gives a message by binding to the site and acting as a modulator. So here you can see that in most of the metabolic pathways you have different enzymes. From A will be converted to B will be converted to C. You have a step or a cascade of reactions which are controlled by different enzymes. For example, enzyme 1 is converted to enzyme 2. Enzyme 2 is converted to enzyme 3 by the substrate B, and finally you have the end product. So this end product itself goes and binds to the the enzyme, and then it actually inhibits the pathway. So this is what is called as a feedback inhibition. So this is more clear. You can see that there is a cascade of reaction. A is converted to B by enzyme 1. B is converted to C by enzyme 2. C is converted to D by enzyme 3. Now, what happens? This continuously it is happening. Okay, for example, synthesis of gluconeogenesis. Continuously, glucose is being synthesized by this step, but body does not require so much of glucose. So, okay, so this is just hypothetically, glucose is not a feedback inhibitor. So here, what happens? So D itself, there is a lot of D. That is why 3 Ds. Okay, so that is going and telling and binding to the first enzyme. That is what is called as a rate limiting enzyme. It goes and binds to it and tells, okay, enough, that is enough. No more further reaction to take place. So this is what is called as feedback inhibition. So here, this is a classic example, conversion of threonine to isoleucine. Okay, so initial reactant is threonine. It undergoes multiple reactions, conversion to A, to B, to C, to D, and finally you get isoleucine. In between, you have different enzymes, enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. But what happens when a lot of isoleucine is being synthesized? This isoleucine itself goes and binds to the rate limiting enzyme, and thus it inhibits, or it is called as the feedback regulation. So that's it for now. So today we were discussing about mainly about the allosteric regulation, and regulation is very, very important, especially in metabolic pathways. And allosteric regulation, if you understand, one thing you should remember is it is by binding at a site other than the active site. Okay, and this happens by molecules called as modulators or effectors. You have positive modulators and you have negative modulators. Positive modulators are or negative modulators for that matter. What do? Is they bind to the site other than the active site, and it changes the conformation of the active site. Thereby, it inhibits the binding of the substrate. Classic example is ATCase and phosphofructokinase, which helps in, which is a main part, which is regulated allosterically during glycolysis. Okay, so thank you.